I told you that the axon is very long, so that leads to some challenges. And in 2016, I'm correct, it should be the reference. My bad, sorry. So this is a paper, a review paper in Neuron by Tom Schwartz and Thomas Misgelt that define a concept that is called mitostasis. Basically, this is asking the question, how do you maintain homeostasis, which is the equilibrium of the cell, in a challenging cellular environment? And so, compared to the soma, if you want to have mitochondria here that are healthy at the extremity of the neuron, you need to be able to control mitochondrial biogenesis, the dynamics of fusion and fission of mitochondria, their distribution, the transport along the axon, and also protein turnover and elimination of mitochondria by mitophagy. So why all those functions are important? Well, it is well known that if you affect mitostasis, if you affect anything that I just described so far, biogenesis, turnover of the protein of the mitochondria, transport of the mitochondria, then you would have altered energy homeostasis, and this has been long described as a hallmark of neurodegenerative diseases. So really, the field of neuroenergetic has been driven by the connection with any neurodegenerative disease that you can think of: Alzheimer's, Huntington's, Parkinson's. Most of them would be connected with the mitochondria. But the other aspect, and the one on which my lab is more working, is what is happening during the development of neural circuits. Before neurons can die of age, there is one step that is crucial, is to organize this dense network of neurons and to make sure that neurons can make those connections, can develop axons that are healthy, make synaptic contacts with the proper neuron through the proper dendrite. And this process is happening during the embryonic development and during the first years of life in humans. And the interesting part is that if you measure glucose and oxygen consumption by the brain, you can see that it peaks at birth. And this is really the time when the brain is forming itself and forming the most functional circuits. So this is where you have the wiring and refinement of neural circuits. So now there is a strong association with altered neuronal metabolism, altered mitochondrial function, and many diseases that affect neurodevelopment that leads to complex phenotypes, including epilepsy, including intellectual disability. So this is a question that my lab is addressing. And now I go maybe into the topic of the day. To address this question, we need tools. We need to be able to measure metabolic activity. So I will describe a bit what are the tools that are at hand and why we choose to use biosensors. Then I will describe a bit the advantages and the limitations of those tools. And then later on, I will tell you a bit more about how we use them in the lab. So unfortunately, I am not very rich into the technical details. I will not give you a step-by-step protocol. One of the reasons is because you need specific microscopes and you need to design your protocol for each biosensor that you will be using. But on the other hand, I will try to give you a general overview. And as anything, I'm always happy to share specific protocols. So if one day you want to use those tools, don't hesitate to contact me. I will always respond and try to help you to set that up. So how do you study metabolism? Well, the first question is, how do you define metabolism? So usually when I give my class, I ask people to participate, but because this would be unfair for people online and because I'm already talking too much, I go straight to the answer. And the answer is actually pretty funny. If you define metabolism are the chemical reactions that support life. So this is not really helpful because basically any reaction in the body is metabolism. It can be more operationally divided into catabolism, which is the destruction of a molecule to produce another molecule. And the catabolism is also a way to produce energy. And the other aspect is anabolism, which is using energy to build molecules. And you always have an equilibrium between the two of them. And also, for the rest of the talk, I will define metabolism more in the sense of energy metabolism. So loosely, how do you produce ATP by the cell, starting from glucose or starting from fatty acids or other type of fuels. So now, these are the metabolic pathways. They have been studied by so many labs in so many fields. We pretty much know all the enzyme reactions that are happening here. So this is very complex. You can see here this circle that you probably remember from the lower classes as the Krebs cycle, because we always start with that one. This is the one that we can recognize. But all those pathways, people dedicate their life to study one single node of metabolic regulation and its connection with the disease. The nice aspect of that is that every single node is enzymatic reaction. So this is driven by a protein, an enzyme, that will have a biological function. So now if you want to study metabolism, since this is all proteins, you can pretty much apply all the techniques that will lead you from gene to molecule. The information that is coded by the genome, so you can do genomics from patients, study of the transcriptome, transcriptomic and bulk or single nuclei RNA sequencing. And then that would tell you how much every single gene is expressed. But from the gene expression to the protein, there is another step. So you can do proteome analysis that will tell you if the protein is actually being made, and also maybe if this protein is being modulated by phosphorylation or by any other post-translational modification. And then you can do metabolome analysis, which is an unbiased way that you will measure all the metabolites that you can detect in the cell, usually by mass spectrometry. So this will give you this type of images where you have pathways and you can measure given metabolites, and then you can see what are the pathways that will be upregulated and downregulated. This one is for the highlight. This was an experiment where we have a strong deficit of mitochondrial function, but no changes into metabolites concentration. And that is the message. There is one limitation in all those methods that are unbiased. The nice thing is that you don't have any assumption. You don't need to know what is wrong. You just have to know that it has to deal with metabolism. And then the analysis will tell you what are the hypotheses that you should go after. Or maybe this is lipid. Maybe this is something to deal with the mitochondria, and so on and so on. This will all come out of the analysis, except that you only see a snapshot of what is happening at one time. You don't have a dynamic information, or it's very hard to have the dynamic information. And in this case, the mitochondria are not working properly, but the cell is very good at maintaining the balance, at maintaining the homeostasis. It's just that when you trigger some sort of stress, it cannot respond anymore. It does not show from the metabolome analysis. So if you want to be more functional, then there are some alternative methods. Historically, focusing on the mitochondria is a good way to produce energy by using oxygen. So it's easy to put an electrode in your assay tube and then to measure changes into oxygen consumption. This is the Clark electrode and some things that you have learned in your physiology classes. Now, this type of thing can be done with several types of analyzers that are commercialized and that come with kits and assays that are defined. So one example is the seahorse that is now commercialized by Agilent, but you can also use or hear about growth technology. They would be providing the same type of information. This is measurement of some changes that happen in your mitochondria or in whole cell populations. In the seahorse, for example, what you can measure is acidification of the medium and the use of oxygen in the medium. And you can see the rate of acidification, which is correlated with lactate released by the cell, so how much they do glycolysis, and how much oxygen they use in mitochondria. And then the idea is that you have commercial assays with this type of machine. You have injectors that automatically will put some drugs that will challenge metabolic pathways, and then you can see how they respond. So with oligomycin, you will inhibit the ATP synthase. So you will block the synthesis of ATP by the mitochondria, and you will see a drop in oxygen consumption, which is indicating how much ATP is being produced by the mitochondria. And conversely, you will inject FCCP, which is a decoupling agent that will negate the transport of proton by the supercomplexes of the mitochondria. So then the mitochondria will start turning like crazy because it cannot make the gradient of proton in your membrane. And then you can see the maximal respiration that can be achieved by the mitochondria. This is all functional information. So this is relatively easy to implement as long as you have the machine. The machine can actually, it's pretty pricey, but it can be a core facility machine because the assay does not take that long. It's very good for cells. It's not that good for tissue because it's designed on 96 well plates. So with tissue, it's a bit more complicated. The disadvantage mostly is that you don't have a single cell resolution. If you want single cell resolution, that's where microscopy really takes center stage. And biosensors is nothing new, actually, since the discovery of fluorescent proteins, such as GFP. Some people have been modifying them and say, okay, what happens if you add some modules to it? This is all proteins, so you can always add some sequences to your DNA that will be translated into your protein. And then you can modify them, and there are some labs that are dedicated that discovered that actually this barrel of GFP, the fluorescence comes from those structures. So if you have an alteration of the structure by the one thing that you add, then it can change the fluorescent properties. And one very classical experiment that you have all heard of is GCAMP, which is the most commonly used biosensor for calcium. So GCAMP is basically GFP molecule that is attached to a module that will bind to calcium. And depending on the binding of calcium, it will just close the barrel or open the barrel. So you express that in your favorite cell, in a neuron, by transfection or by electroporation. This is an easy step. This is all based on plasmids. And then you can, with a single microscope, detect GFP fluorescence, green fluorescence. So pretty much all the microscopes can do that. And when you have a change in cellular conditions, such as action potential, you can see a peak of calcium in your cell. And you can see that at the whole cell level, but you can also have resolution of what is happening at the synapse specifically. So this is one of the most classical examples, but the same method works for biosensors for any metabolic pathway, biosensor for signaling as well. You can see how much enzymes, kinases, and so on are activated or inhibited using the same type of biosensors. And now this is used in vitro, but this can also be used in vivo because this GCAMP or this metabolic biosensor, you can put that in an adenovirus, and then you can inject that by stereotaxy into the brain. And with 2-photon microscopy, you can measure changes into fluorescence in the brain. So you can see whatever biological function you want to see in the live brain, in the live animal. So this is very nice. And there are several modalities now that are used for those biosensors. So I will give you one example and maybe try to describe why some are maybe better than the others. ATP measurement in the cell is something that has been developed since 2009 with 2 biosensors. One is from a Japanese group, and this is based on fluorescence transfer by FRET. So I don't know if you are all familiar with FRET. The idea is that any fluorescent molecule has one excitation peak. You will put some light into it, and by the energy that is provided by the light, it will turn your fluorescent molecule into an excited state. And the release of this excited state return to normal will emit a photon that is the fluorescence that you are actually looking at. All right. Just let me know if that is not clear. So the idea is that you give energy, you give light energy to a fluorescent molecule. It will give back some light energy, slightly lower, and so the more energetic is the blue, the less energetic is the yellow and the red, which is why the blue will be lit with a 435 nanometer laser and will emit into the 475 nanometer. The nice thing is that those fluorescent molecules, they can actually communicate with one another, because light that is emitted by this molecule is more energetic than the light that you need to turn on a yellow fluorescent molecule. So the whole principle of red is that if the two molecules are close enough, then some part of the energy that is released by the cyan fluorescent protein will be caught by the yellow fluorescent protein and being used to emit some yellow light. Okay? So this is something that is purely dependent on the physical distance between the two molecules. And why this is important? It's because you can have those, by the way, I didn't go into the details, but CFP, YFP, GFP, and so on, they are all based on the same protein structure forming those barrels. You can move from one color to the other by just mutating a few amino acids. This is basically the idea. So this is all protein. So you can have these proteins in cDNA and just put a linker between them two. And if this linker is capable to bind some molecule, then the change of conformation will induce the two fluorescent proteins to get close to one another. So now we have this biosensor that is binding to ATP. When ATP is bound, there is the closed state, and it will emit the FRET fluorescence. So this is one modality. One other modality that was developed at the time is actually based on one single GFP molecule that is attached to a module that will slightly twist the GFP. And this is depending on if this module is attached to ATP or ADP. So this one was developed by a group of Gary in 2009 as well. And basically the idea is that by just changing slightly the conformation of the GFP, it will not change the emission. It still emits the green fluorescence. So you can still detect that with your favorite green fluorescent microscope. But it will change how much it can be excited by different wavelengths. So you will actually have a microscope that will turn the light at 405 nanometer and 480 nanometer. You illuminate with two different wavelengths, measure the GFP emission, measure the green emission. And in the conditions that are rich in ATP, it will not be that much excited at 405, but really much excited at 480. And if you have low ATP but high ADP, you would actually be the black curve, so you get more excited with the deep wavelengths and less excited with the higher wavelengths. So you just do two pictures of the same cell, and then you can detect how much, you can quantify how much ATP and ADP there is in the cell. Okay? So now why two different modalities? Well, one of the limitations is that for that you need a microscope that is able to read the green with a decoupling of what is the excitation for the green. And not all the microscope can do that. There is a very standard excitation light for GFP. So if your microscope can only excite with this wavelength, then you cannot use this type of rational probe. But the advantage of this one is that it's very easy to detect the green. Whereas to detect cyan fluorescent protein and yellow fluorescent protein, then you need specific lasers that not all confocal would have. So depending on what is your microscope, then you would select one or the other. One other limitation is that the FRET is usually very low efficiency. when you measure the fluorescence, maybe 90% of the signal that you will measure is just the fluorescence of the yellow fluorescent protein, and only 10% of that is the transfer of energy from the cyan fluorescent protein. So you have a signal that is 90% noise, and only 10% of that is the thing that you want to measure. So you need to have some changes that are very high in the cell so that you can detect them. So FRET is much harder to master, and it needs some computation that is sometimes a bit. So really, depending on what type of microscope you will have, you will pick one or the other. Or the third method is actually based now on circularly permuted GFP. So it's like GCAP, right? You have one fluorescent protein based on the GFP cytoskeleton that is in open conformation, and then when it binds to ATP, it closes and it will emit the light. So this is basically the opposite of Perceval, where there is no fluorescence, and then with ATP, there will be fluorescence. So you will see some changes in emission, but no changes in the excitation. This one is by far the easiest to manipulate. If you can measure calcium, you can measure ATP with that. The drawback is that this one is only one fluorescence, whereas with this, you have 2 lines of fluorescence, and with this, you have 2 lines of fluorescence. So you need one of the color, you need some red, to be ratiometric and to normalize how much fluorescence you have in your cells, how much transcription you have in your cell. I will comment on that in 1 minute. Okay, I hope it was not too much detail. Now I will go a bit more into what we do in the lab. So the step is actually fairly simple. It's always based on the same. You do electroporation of embryonic brains. This is the way that you can get some plasmid to go into progenitors and then being expressed into neurons. Either you let the mice live and then the neurons will be integrated in the brain in vivo, and you can do imaging in vivo, or you just collect those embryos, do a dissociation of the embryonic brain, and do some culture of neurons in vitro. This is very classical, like pretty much all the labs during neuroscience can do this type of experiment. This is the basic neuronal culture. And then you use a microscope that will be adapted to the biosensor that you want to use. So you will always find a microscope next to you in your core facility, in your lab, or in the lab next door. That can allow you to do this type of experiment. The real limitation is that you need to have the proper laser lights that are adapted to your biosensor, or you just change the biosensor. And the other thing is that metabolic activity is dependent on the temperature. So you need to be in an environment that is controlled by the temperature. You need to be at 37 degrees. So it just doesn't work. So you need this whole big box above the microscope that will maintain the temperature at 37 and the CO2 at 5%, just like in an incubator. And the last thing, and because you are doing some live experiments, you need an inverted microscope. You need the light to come from below and not from above, because you cannot just flip the box. The cells will be in a medium that is adapted to imaging, so a medium that does not contain some phenol red and other stuff. Anyway, once you have that, then you can select the probes that would be adapted, and you pick each probe on every single metabolic pathway or every single metabolite that you want to measure. So in my team, we use measurement for ATP, we use measurement for pyruvate, for lactate, for the ratio between pyruvate and lactate, for the ratio between ATP and ATP. Also, some probes that will measure pH and the membrane potential of the mitochondria, as well as some probes that will measure the activity of signaling enzymes. And then you can measure metabolic activity in the live cell and measure metabolic fluxes. And in our case, we adapt that to measuring what is happening in terms of metabolic regulation in the development. Okay, so that was long. That should be shorter. What are the advantages and disadvantages of those methods? I see many advantages, but this one I want to show to you. And for once, I did not forget the reference. I actually put it twice because you have the paper and you have also the name of the guy, Journal Bex, fantastic researcher. He has made one paper that basically changed my vision of what is happening in the cell. I assume that most of you must have done Western blots and then see burns increasing or decreasing, depending on, oh, okay, this protein gets phosphorylated. So we see the phospho signal that is increasing. And that seems to be like the end of the story. But this is actually an average of what is happening in all the cells that you are looking at the same time. If you look at things one cell at a time, then it changes radically. So this is one example. This guy is using John Albeck. He's using biosensor to measure kinase activity for specific signaling pathways that are important for cancer. This one is AMPK. This is a biosensor for metabolic activity. And the more orange red, it will be more active, and white is inactive. Now, AMPK is responding to metabolic challenges. So if you put an inhibitor of glycolysis, then you will see the signal getting more red. And he will put the inhibitor here, but what you see is that actually the signal is not becoming red in all the cells at the same time. And also it's not staying red. Some cells will be pretty stable. Some cells will have those oscillations that you can actually measure one cell at a time, and you can see oscillations that are not always synchronous between all those cells. And depending on the quantity of drug that you see, you will see that some cells respond and some cells do not respond. So sure, when you do average, this is exactly what you do with the Western blot. This is what you do when you measure all the cells. You would see this nice response, but then one cell at a time, this is actually different. And why this matters, why these oscillations are actually functionally important? Well, depending on what type of metabolic stress you are using, if you use inhibitor directly for the mitochondria, then you will see that not exactly the same. Similarly, you would have activation of AMPK, and you would see single cell activation, not all the cells respond the same, but now on a more slow pace, and you would have some cycles of activation and inactivations that are happening differently. So this is the type of oscillations that you will be seeing. And then the same, if you do averaging, you see an increase. So if you do the average, you would have the feeling that both conditions, both drugs, they induce the same response. Whereas at the single cell level, this is radically different. So this is one of the very powerful advantage of using biosensors. It's that you can address what is happening one cell at a time, and even at a subcellular resolution. You can see one synapse at a time, even. One other advantage is that you have a dynamic picture of what is happening. This was one image of the use of this ATP-ADP biosensor perceval into the axon. I showed that to you before, and I described that there are hotspots of metabolic activity. But the idea is that this is pretty much the same as with metabolomy. You see on this picture at a given time what are the equilibriums of ATP production in the cell. But the steady state level of a given metabolite, ATP like anything else, is basically the sum of the production minus the sum of the consumption. And those evolve with time and depend on the conditions that the cell is subjected to. So if you want to see not only the level of ATP, which the cell will try to maintain constant, it does not want to be lacking energy, so it will do its best to maintain the ATP. What really matters is how capable it is to produce ATP. What are the fluxes, the metabolic fluxes that you can have, right? If you want to measure glycolysis flux, for example, it's not sufficient to measure how much glucose and how much pyruvate you have at a time. You need to see how fast the glucose is converted into pyruvate. Same for ATP synthesis. So basically, the idea is that with those biosensors, you can put a drug on them, and then with an inhibitor of the mitochondria, you can see extension of the signal and measure the speed of extension of the signal. And by just measuring here the speed by this linear phase, you can get any idea from one cell to the other of what is the rate of consumption of ATP. And you can see that in wild-type neurons, in the axons, it is fairly constant, regardless of what is the start point. The rate of consumption of ATP will be similar. But if you look at now wild-type versus a knockout condition, then you would maybe see some changes in how much ATP they can produce or how much ATP they can use. All right, so this dynamic view is also the second very important benefit of those biosensors. Now, those have general limitations. They are not the perfect tool, and actually the idea is that when you go into metabolism, you need to combine strategies. So one very general limitation is that as for everything that is based on light imaging by microscopy, you have some general limitation. One important is the dynamic range, right? You want to be sure that those changes into fluorescence that you see, if you see twice as much fluorescence, does it mean that you have twice as much metabolite or not? So this is really dependent on the dynamic range of your biosensor and what is its Km, how much affinity it has to bind to the metabolite that you want. If the biosensor is saturated, if 99% of the biosensor is bound to what you want to measure at the physiological level, then the changes that you will see in the cell would not induce some significant changes in fluorescence. So all those probes have well-described papers that define what are the Km, and depending on what you want to see, if you want to see something at the intracellular level, at the extracellular level, in some cell versus some other cell types, then you need to pick the right biosensor. There is also phototoxicity that can be induced. So if you start to image for too long, then the cells won't like that. So you always have to find a good compromise between high sampling rate. You want to image with a high frequency so that you see events that are happening in real time, but also the faster you image, the shorter the whole image session will be. So if you want to see things that are happening on a long scale, then you need to make intervals and to image with less frequency. Biosensors are influenced by the cellular environment, and one very well-characterized thing is that most of them will be sensitive to pH. The threat biosensors are actually less sensitive to pH, but if you think of flexible, which is widely used, if you have a tiny change in pH in the cell, then you would see some responses in terms of fluorescence that are massively different. So most of the time, you actually need to measure pH with another biosensor in real time and do some complicated calculation so that you can make sure that the effect that you see metabolic activity and not due to change acidification of the cell. The binding of the ligands to your biosensor could also, in theory, affect cell signaling. Right? If you use GCAMP, you want to be sure that GCAMP is not taking all the calcium of the cell. Otherwise, the cell will start to respond abnormally, not because it is abnormal, but because you overexpress too much of an artificial protein that is capturing the calcium. So this is the same with metabolic biosensors. If you bind all the ATP, then the cell gets into metabolic stress, just because you overexpress a protein and not because it is in a mutant condition or because the drug that you put on it is stressing the cell. So this is things to consider. And then signal might depend upon expression, how much plasmid there is into the cell. If you do transient expression, which is what we do in neurons, then some neurons will have 10 copies of the plasmid. Some neurons will have 100 copies of the plasmid. So of course, the signal that you will see will depend largely on how much protein is expressed. And this is where proteins that are based on cyclic fermented GFP are tricky to use because basically you have no signal, and then when you induce your stimulus, you will see this peak of signal, and it will go as high as it can, depending on how many molecules you have. So depending on the transduction efficiency as well, which is why you always need to have a co-transduction with some other molecules, such as a red fluorescent molecule, that is not dependent on metabolic activity at all, but that will serve as a normalization on how much transduction efficiency you had in the system. All good? All right. So I'll try to give you, for the last part, a few examples on why and how we use them in our neurons. Hopefully, it will be more basic and down to earth. So my lab has been interested since I was a postdoc in kinases. One that is called LKB1. LKB1 is a tumor suppressor gene that has been used, that has been studied as overall regulator of the AMPK and other metabolic enzymes. It induces some signaling pathways that are important for metabolic regulation in the cell, and also for polarity of the cell. It's something that is often deregulated in cancers. So when I was in postdoc, I started to address the question, what is LKB1 doing in neurons? And we identified that LKB1 and MUAC1, another kinase that is working downstream, both have a phenotype that is the control of axon branching and the formation of those collaterals. So it works in vitro, it works in vivo. But the real question was, what is the biological function that supports, that makes the link between those kinase and axon branch? So long story short, at some point we got interested into how much this pathway could be influencing the transport of cargo into the axon. And our hypothesis at the time, that was wrong, but that still led us to discovery by chance, was that maybe there would be some mix-up between dendritic proteins and axonal proteins, and if dendritic proteins are by accident transported into the axon, the transport of axonal protein and axonal cargo is not happening, then that might be the development of the axon. So the axon is like this highway where you have rails of microtubules and then trains of cargos, vesicles, mitochondria, other organelles that just go along based on molecular motors of the kinesin and dynein findings. So how do you do that? Well, you basically get fluorescent protein, red fluorescent protein, green fluorescent protein, that is tagged to your favorite organelle with a peptide sequence that is an addressing sequence. So we have a mitochondria targeted GFP that is expressed in neurons, and then those are axons in wild-type conditions and in knockout conditions, where every single dot that you see is a single mitochondria. So now the funny thing was the dynamic part. And before I start the movies, I want to say what is known from the literature. If you look at mitochondria in immature neurons, 5 to 7 days in vitro, roughly 70% of them, 65–70% of them, would be fairly stationary. They are at a given position and they would not move too much over the 30 minutes that it takes for us to do the movies. And then 30% of the mitochondria would be moving and mostly directed toward the extremity of the axon. So this is what you would see here, that those mitochondria here, they sort of handle locally. And then from time to time, you see one that crosses the screen. The obvious thing that we saw in the knockout condition was that mitochondria, they are moving all the time, and they actually lost the capacity to connect to the axon. We by that if you inhibit LKB1 on MUAC1, you decrease the percentage of stationary mitochondria. And then if you overexpress those kinases, you would increase the stationary mitochondria. And so that was the start of the story that led to me and my postdoc and to the creation of my lab in Lyon. One is, where do those mitochondria stop? Right? If you have a signaling pathway whose function is to support the stopping of mitochondria in the axon, it's obviously very critical that they stop somewhere. The cell puts a lot of effort to arrest those mitochondria. So where do you want mitochondria? And the idea is that we thought that it would be the synapse. So we made some multiplexing, the mitochondria in red and the synaptic vesicles, so the presynapse is in green. And with that, you can do this dual imaging, exactly the same, where synaptic buttons will be in green and mitochondria will be in red. And you can see the motility of those mitochondria going from one synapse to the other, and so on and so on. So we could quantify that. These are some kymograph views. So on the horizontal axis, you see the axon that has been linearized by the computer. And on the vertical axis, you see every single frame of the movie that forms an additional line. So when you have a vertical line here in green, that means that the spot is fixed into the axon. This spot will give a vertical line because every single frame you would see the same signal at the same position. Whereas those mitochondria that are moving, like this one, every single frame would be going a bit further. So you would have those diagonal lines. And then the idea was that if you inhibit LKB1 on UAC1, the mitochondria and the presynapse do not overlap. Whereas if you overexpress them, not only the mitochondria are not moving, but also they are stopping specifically at presynapses. Okay, so there is a correlation between bringing mitochondria and stabilizing axon ramifications. How does it work? And the idea is that through the literature, there are two modalities for making collaterals. One is something that I would call the branch comes first model. Basically, you would form collaterals. And then those collaterals would meet with a postsynaptic termination, form a synapse, and this synapse is stabilizing the ensemble, and then you jump from one synapse to the other. In this case, you first form the branches, and then you form synapses. There is another modality, mostly from peripheral nervous system, where it has been described that the mitochondria would at some point stop along the axon and then induce some local translation of cytoskeletal proteins such as actin. And this local translation is inducing the conversion of philopodia into branches. So in this case, this would be a branch comes last because you first bring the mitochondria and then you form the branches. So which one is which in our cortical neurons? And for that, my PhD student, she looked at the dynamics of mitochondria into the axons, but she wanted to see that over very long periods of time because she wanted to be able to see spontaneously the formation of those collaterals of the axons. And this is something that happens on a time scale of 24 hours because we don't know where it will happen. We can only see that there is an axon here, but where will a new branch form? We have no idea. So we have to maybe hope that over 24 hours, we can see spontaneously formation of collaterals. So the idea is that she has to design some high frequency, high pulse imaging, where within 5 minutes, she can see the dynamics of mitochondrial recruitment with some pause time where you can actually let the axon rest, decrease the phototoxicity, and so it continues to develop. And this is one example where you would see the branch form here, a few mitochondria that are around the place. But those mitochondria don't seem to be very interested. They start leaving the zone, and this branch is not stabilized, it retracts. Whereas in this condition, again, you see formation of a branch. And here, not only the mitochondria accumulate at the base of the branch, but also they can enter into the branch. And in this case, the branch continues to grow and keeps on being here. So this, we could quantify that the dwell time of mitochondria at the origin of the branch is higher than along the axon, and that when the mitochondria enter the branch, then the lifetime of the branch is increased. So this led us to the conclusion that mitochondria are important for the stabilization of the branches, and that in cortical neurons, this is actually the first model that is happening. The branch comes first, make pre-synapses, and then mitochondria are recruited later on for the stabilization rather than for the formation of the branches. Can you prove that? If you want to prove that, you must be not only observational, but also have an intervention into the system. And for that, some biosensors that we were using are actually modulating mitochondrial activity. So this is based on the expression of a protein that is called killer red. Killer red, despite having a cool name, is just a red fluorescent protein that has a couple of mutations that will make it not very efficient at emitting light, but very efficient at converting the energy into the production of ROS. So now if you address that to the mitochondria, instead of making some light, it will make some ROS that will degrade the mitochondria. And our idea was that if we can degrade mitochondria locally, then we would predict that we would lose the stabilization of those branches. This is the proof of principle. Those killer red are expressed in HeLa cells, where you can see this network of mitochondria. And this biosensor is a biosensor for loss production. So when you illuminate, you can see that it will very rapidly, within 30 minutes, turn red. And also you will see that those mitochondria start to fragment. They start to get round, and they have signs of poor health. That is indicating to us that we are successful into degrading mitochondrial function. And the nice thing is that it only happens in the region that we illuminate. So we have a nice spatial control over what is happening. So now we can go into neurons and then select a branch, destroy the mitochondria locally, and see what happens. And within 15 hours, you can see that this branch will retract where we remove the mitochondria. And this is not because the neuron is dying, because you can see that all along the axon, you can see more formation of branches. So we could quantify that. This has been published recently, that in the region of interest, we trigger retraction of branches when we can remove the mitochondria. So the next step is those mitochondria that are so important for the stabilization of the branches, what are their function? Again, mitochondria, they are important for generation, for many metabolites generation, but also for the regulation of calcium and also for signaling events. So we got interested into metabolic regulation. One of the reasons is that LKB1 and UAP1 are actually important for metabolism in cancer cells. So we wanted to see if that was the answer as well. Mitochondrial metabolic activity was the answer in the stabilization of collateral branches of the axon. And for that, I described previously how we can use those flux analyzer to measure metabolic activity. So if you remember the Seahorse assay, you can measure glycolysis and you can measure mitochondrial respiration. And then in gray, you have wild-type neuron. In red, you have knockout neurons. And what was obvious is that the gray and red curves are the same if you look at glycolysis. But if you look at mitochondrial respiration, there is a massive decrease of the basal and of the maximal respiration in those knockout conditions. So those neurons that are deficient for LKB1 and for UAP1, they have a lower capacity of using their mitochondria for metabolic purposes. Why the connection with axon? Then we use biosensors. We use Perceval, where you can see what is happening at the scale of the axon, and you can couple that with knockout neurons, and you compare, and then you would see that not only you have a reduction of ATP-ADP ratio in the stoma, but also specifically something like a currency of a lack of ATP is happening in the axon. And again, one of the beauty of those biosensors that are genetically encoded is that because everything is based on DNA converting to protein, you can add whatever addressing sequence you want in front of your protein. So you can put those biosensors with mitochondrial targeting sequence and look at single mitochondria one at a time. So we've done that using this red sensor for the mitochondria, and you see dots of mitochondria that can respond to peroxide and get more oxidized. So you can compare the growth production by the mitochondria in wild type and in knockout animals. And the same, you can use dye that will be targeted to the mitochondrial matrix based on its proton charge, so based on its capacity to synthesize ATP. And so, long story short, with those, you measure plenty of mitochondria and plenty of axons, and you do some cumulative frequency curves that demonstrate that there is a decrease of mitochondrial membrane potential and an increase of growth production by those mitochondria. And this experiment, the thing that I forgot to say, this is important for us. Why? It's because I told you that in knockouts, mitochondria keep moving all the time. So moving using molecular motors is something that is using ATP, right? So we wanted to be sure that those mitochondria are actually defective and not that they work normally, but it's just that because molecular motors are so much upregulated that the ATP that is produced is immediately being used by those molecular motors. With this type of approach, what we can tell is that single mitochondria, rather moving or not, are actually less efficient at producing ATP. And one last thing, again, you want to be interventionist, not just make some correlation. So what would happen in knockout condition where mitochondria are less efficient, they work less, if you could boost mitochondrial activity? And for that, we use a drug that is acetyl L-carnitine, which is helping the mitochondria to use fatty acids. So if more fatty acids can enter into the mitochondria, then it can in theory produce more energy. And the prediction would be that you can rescue the collaterals that are being formed. And it actually worked. These are some neurons that are knockout from your acquired. So remember, I told you that you have less collaterals that are being formed and that you have less mitochondrial potential in the axon. Now, if we treat with L-carnitine, not only we can rescue membrane potential of the mitochondria, so we can rescue mitochondrial function, but we can also rescue the formation of axon collaterals. So with that, I will leave you. Thank you for all of you for being in the room and for all of them, all of you who are online. This is a picture of my city of Lyon. It's a beautiful city for everyone who wants to visit, but it's also a nice city for doing science. So if you are ever interested, especially in a postdoc or a PhD, I'm always looking for candidates. I also want to emphasize that a lot of this work has been funded by the European Commission, the ERC, and the Marie Curie Action. And this is something that is important to my heart. I've done my PhD in America, but I choose to go back to Europe, and I think that Europe has many things to offer. So this is why the invitation to attend to the Browning seminar was a very special occasion for me, because I'm happy that I can communicate with so many European countries at once. And I strongly encourage all of you to take these opportunities to travel and to visit Europe, because that can only open your mind.